I have to admit, I just had two thoughts. One is the incredible honor it is to follow one of your personal heroes, a titan of philanthropy, a visionary, and one of the most articulate spokespeople for the Institute. I can think of Lester Crown, and then there's the problem, which is you're following Lester Crown. So anyhow, good evening. Welcome to the 2019 Leadership Retreat, an annual meeting of the American Committee for the Weizmann Institute of Science, and thank you so much for being here this evening. Let's take a moment to wish ourselves a happy anniversary and celebrate a small group of visionary leaders who, 75 years ago, rallied like-minded Americans in support of a modest research facility in pre-state Israel. 75 years that have seen the Weizmann Institute transform into a globally renowned powerhouse of science, research, and education. 75 years of institute-developed drugs, products, science discoveries, and ideas changing and saving lives worldwide, and 75 years of American philanthropy partnering with Weizmann scientists to transform tomorrow. And like our founders in 1944, who also came together for leadership meetings in Boston and New York, as we do tonight and tomorrow, we have the opportunity to effect transformative advances, not just for the Institute, but for everyone, everywhere. This evening, we learn firsthand of the transformative power of philanthropy and life-saving results. Our serendipitous story begins in the early 1990s at the Weizmann Institute, when the late Professor Yoram Solomon of the Biological Regulation Department and Professor Avigdor Schertz of the Plant Sciences Department had a friendly, casual conversation on campus. We'll hear more about this in a few minutes. This chat between a chemist and a plant scientist is the ultimate example of what the Weizmann Institute does best: multidisciplinary collaboration, fueled by curious minds. 2 decades later, although it was not their original intent, these two gifted scientists became inventors of a remarkable cancer therapy. Along the way, the plot developed to include a pharmaceutical company that collaborated on a highly innovative yet risky idea, a partnership with international hospitals and world-leading physicians, including Dr. Jonathan Coleman of Memorial Sloan Kettering Cancer Center, and passionate philanthropists who believed in the research and in the scientists doing it. The late Alan Siegel, our dear friend, and then director of the Thompson Family Foundation, met Professor Schertz in 2011 and heard about plans for a groundbreaking collaboration with Memorial Sloan Kettering to advance research in prostate cancer. Alan had the wisdom and vision to recognize the enormous potential of the relationship between two of the world's greatest institutions, and by channeling Wade Thompson's desire and commitment to fighting all cancers, Alan was able to see this research come to reality. The Thompson Family Foundation's multi-million-dollar grants enabled clinical trials to get up and running relatively quickly at Sloan Kettering. The foundation's transformative philanthropy supported bench-side biomedical research and the resulting translation to clinical trials. It enabled progress on a shorter timeline than usual, and therefore directly benefited cancer patients more quickly. That visionary philanthropic support continues to this day. Now, I have the privilege of acknowledging and thanking two very special people who can't be with us tonight. First, Sandy Siegel, Alan's wife. Sandy, thank you so much for being with us. You will always be part of the Weizmann Institute family. Alan will always have a special place in our hearts. Next, Amy Houston, who now directs the Thompson Family Foundation and ensures Wade Thompson's vision endures. Amy, thank you for joining us and for guiding the foundation as it continues its visionary investments in historical preservation, human services, medicine, and health. We look forward to strengthening our partnership in the coming years. We'll get to the fascinating science and clinical updates in a minute, I promise. But what we should acknowledge and remember first is that this story about the Thompson Family Foundation, Memorial Sloan Kettering, and the Weizmann Institute is part of a continuum of transformational philanthropy that has been the touchstone of the American Committee's inspiration for the past 75 years. We have made a real humanitarian impact because of the generosity of foundations and individuals who also had the long-term vision to invest wisely and had the belief in the excellence of our scientists. They understood that fundamental science, the foundation of the Weizmann Institute, answers the deepest, most essential questions we have about the world and leads to wide-ranging benefits that improve the quality of our lives. Ladies and gentlemen, let's recommit to working together to ensure that we have many more such stories to tell. Now, I'm delighted to introduce two amazing scientists and clinicians. First, Avigdor Scherz of the Weizmann Institute's Department of Plant and Environmental Sciences, who holds the Robert and Yadel Sclair Professorial Chair in Biochemistry. Please join me, Avigdor. And his colleague and partner, Dr. Jonathan Coleman, urologic surgeon at Memorial Sloan Kettering Cancer Center. Jonathan. I thought we would just start with the basics. So if you could spend maybe 2 or 3 minutes telling us just a little bit about yourself and how you found yourself doing what you do. So Jonathan, would you like to begin?
So that's a great question. I actually started off as an undergraduate as a psychology major. So go figure how I ended up in medicine. But that interest in psychology transformed into an interest in psychiatry and an interest in the underlying basis of neurologic disorders and so forth. And as I entered into medical school and began exploring what it was like to be a doctor, I think what really I think changes most of us. I think all of us have this same story, and it's the story of this partnership, and that is that your life takes unexpected turns depending on who you run into. And I tell my kids life is kind of like a big Plinko game, the game where you hit the ball and it goes up and you bounce off and you go in different directions. And a lot of that transformative change comes from who you run into, who your mentors are, who affects your lives in very profound ways. I guess my voice is going in and out, so I'll use the microphone. But that's how my life has kind of changed. And so as I went through medical school, I suddenly met a tremendous mentor and a physician named Derek Vaughan. He was the chairman of urology at Cornell. He convinced me to change my life and to think about urology as a field, and that led me into joining his department. I was going on a completely different trajectory. I was going into neuroscience and neurosurgery, and suddenly I found myself interested in urology. Someone took an interest in my life, and I took an interest in that field. And then my life changed again when I met a man named Peter Scardino, who was the chairman of surgery at Memorial Sloan Kettering, who convinced me to go into urologic oncology. And then my life changed again when I happened to meet Avigdor Scherz, and suddenly I was thrown into a completely different part of my life. And all of these interactions, I think we all have the similar types of stories to explain, and a lot of that comes from personal interaction. It doesn't come from anything else. It has to come from that type of that very close bond that comes from working closely together, meeting interesting people and finding shared experiences and starting a journey together. Avigdor.
Yeah. Okay, so I think that cause and causality play a major role in our involvement. That's a term that I copied from Paul Auster, with whom we had the pleasure to meet about 10 years ago in Israel. We were a bunch of scientists and neurologists that he wanted to meet, and we spent like 3 or 4 hours with him talking about many things. So I started as a physicist and chemist, actually, and specialized in photosynthesis, and in particular developing in the last years that I was mostly doing it, new ways to make photosynthetic organisms live in higher temperatures. So much before we found out that indeed the change of 1 or 2 degrees can make a big change in the photosynthetic productivity. And then talking about cause and causality, we had a cancer in the family, and this turned everything on. And I thought, well, maybe there are some other ways to take care of cancers rather than, harsh chemotherapy, radiotherapy, which have success in many cases, but not usually. And then it turned out that together with Yoram Salomon and based on my experience in photosynthesis, I thought, well, how does nature get rid of malfunctioning organs? And defining the tumor as a malfunction organ, we look for ways to do it. And the arsenal that I had was the arsenal from photosynthesis. It was chlorophyll, bacterial chlorophyll, light, excitation, and so forth. And as it was said before, I knew Yoram actually as the big brother of my inmate, my friend in high school. And I met him in the corridor of the Weizmann Institute, and I asked him whether he had some compounds that we can hook to our bacterial chlorophylls in order to try to target to melanoma cells and to do something with it. And that's how it started, and the rest is history. And then during the years, we were based in the basic sciences. And the moment of meeting Alan, that was meant to be 15 minutes, right? I was on my way back to Israel in the middle of a pig's experiment that was with one of the fellows of Jonathan. And I said, I don't have more than 15 minutes. I told it to Elaine and Eden. And then I sat with this guy, and it was on and on and on and on. And at the end, he asked me, so how much money do you need? I said, there is no need for any money if you're not giving us, and together with Memorial Sloan Kettering, to make these things for real. And I don't know if Jonathan remember, I called him a few days after from Israel. Jonathan, did you get a letter and some email from Alan Siegel from the Thompson Foundation? He said, well, I got something, but I don't know what is the meaning of it. And I said, well, call him immediately. I'm coming in January. We need to talk. And that's it. I mean, there all the people came, and we decided that we can bypass many of the regular obstacles by doing a direct collaboration. And the principle there, as I explained around the table now, was to find somebody that you can really become friend with. And I must tell you, two things happened to me. That's the meeting with Alan, the meeting with Jonathan Coleman. And so friendship and working very hard together. Jonathan established a wonderful group in MSK that took quite a few of our ideas and make them real, and now into clinical trials that he will tell about, and of course realization, hopefully in the FDA, soon to come. And so I think that we outlined, together with the Wade Thompson Family Foundation, with the great help of Amanda and the family there, and recognition of what we are doing, we profiled and outlay entirely new way of advancing very rapidly translation of research from the basic into the clinic. And that's where we are standing at. I think Jonathan can give some more details on the clinic.
I think the big question that all of us really have is, where are we in terms of basic science today, and where are we in terms of the clinical trials?
So would you like to start, Jonathan? And
Well,
then I'll continue.
Okay, well, we're working here. Okay, so let me backtrack just for a moment and say something that a little bit about this story and leading into what you're asking. And so when Avigdor called me about this issue and said I met with somebody who's interested in helping us with the process, and at this point, we knew what we had. We knew that this was something that was special, and it was all thanks to the work that he and Yoram had done together. And we looked at the whole field when Avigdor and I met, and we talked about what we could do with this therapy. And I said, Avigdor, this goes far beyond prostate cancer. And he said, I'm gonna do my imitation. I know. He said, I know. He said, we can, I said, we can, breast cancer, and I said, esophageal cancer, and he said, lung cancer. I said, bladder cancer, and so we said this is much more, and we have a lot to do here. But the process, you all have to understand the way that the process works. For those of us in academic medicine, the way that this process works is piecemeal. The way that we get from point A to point F, or Z at the end, is through little tiny steps along the way. And each of those steps requires that we have to form a little tiny plan and get funding for that little tiny step. And then the next step is going to be a little tiny plan and another little tiny step and another little tiny plan and a little tiny step. And all of this adds up. And that's the traditional process of how this works. And it takes forever for that process to play out because we were applying for grants and going through DOD and NCI and other types of grants that we have at Memorial. We're very lucky. We have a lot of institutional grants, but the bottom line is that these are very small little funding processes, and they all help. They all add up. They are all very incredibly important, and they do a great job. But when Avigdor came to me and said, I spoke to somebody, and he asked us for the picture from A to Z. And he said, we might want to do this for you. And so then we said, oh, if we're going to do that, then that allows us to bypass B, C, D, E, and F and go right to G, and then we can go from there to here, to here, to here. And we painted the entire picture and said we can do all of this. And we can do it in a way that is going to be incredibly efficient because we're going to do the same thing. We're going to do it in 4 different cancers, and we're going to do it all in parallel at the same time. And that allows us to now make maximal efficiency out of everything that we're going to do. And so we came to them and we said, here it is. And they said, fine, do it. And here we are. And we started that 7 years ago. That timeline for 4 different cancers would normally take about 20 years to complete because you have to start with the first cancer, and then the next one, the next one. Now we did them all 4 in parallel. And here we are now. We have trials in prostate cancer, esophageal cancer, bladder cancer, and upper tract urothelial carcinoma. And they're all running simultaneously, and they're all answering the same questions simultaneously at the same time. And for somebody who's a clinician, my job is to take care of patients, it's incredibly rewarding to be able to see all of that happening at the same time and to see the grateful patients who are coming in and saying, you did something for me that nobody else was able to do. And where did this come from? And I can just simply, point them to Avigdor's direction.
Well,
and he comes to my clinic sometimes, and he actually meets with some of the patients on occasion. And it's incredible to see the interaction, and I think from his perspective, as somebody who started off, working in a place like the Weizmann now, to be sitting there in front of patients who are benefiting from this, and for the foundation also to see just how far we've come and how quickly, it's really gratifying. And I'd say it doesn't happen, without the support of stuff like this. I mean, without that, we would still be back at point C. We would still be there.
Well, first of all, I agree, of course, with every word. But I need to add 2 more things. Number 1, on the mode of collaboration. Number 2, on the role of philanthropy. Mode of collaboration, I think one of the new things that we put in is to take the best physicians in the world, excuse me, Jonathan, clinicians, not necessarily MD, PhD, people that know exactly the patients, and ask them to enter into the very early preclinical stage that we are running. So Jonathan is a little bit modest, I must say. I mean, this is his nature. But his group now at Memorial Sloan Kettering is really part of the very basic research, of the preclinical research. Now, what does it mean? It means that when a doctor that finishes fellowship in Memorial Sloan Kettering and need to write a clinical trial protocol, knows everything about the method, participated in developing the method. He does not have to wait for years until papers will come out and tell him what it is. And then this is part of shortening the time. As for the philanthropy, unlike governmental agencies that have this competition between different scientists that usually do not collaborate. I mean, many times you have competition on the same 1 million or 2 million grants, which may lead to nowhere. And we were sitting from the head of this Cancer Friends organization that told us about $160 million that went into lung cancer with no results at the end of the day after 6 years. And here we are with $25 million, and we are in 4 clinical trials that we hope, or 3 now and possibly 2 more in the very near future, that leads to real therapy. How did it come? And these were the tricks. So first of all, once you have this philanthropic agency, they do not need to be obliged to equality, to all these kind of terms. They say, that's what we want. We want to enforce you in a way to collaborate. And that's what happened with the pancreas cancer, the other program that we are running. They took investigators from Cornell, from Cold Spring Harbor, from the Weizmann, from Memorial Sloan Kettering Cancers, where we are PIs there, and they opened the door to collaboration that never happened before. People are opening their labs before they publish the results. Now, all of a sudden, you have for the same amount of money, twice, 3 times, 4 times more synergistic efforts that you could have when you let the governmental agency support it. So there's so many things additional to this model, part of which is that if there will be royalties, they will come back, pay to the foundation, but then the foundation will invest it back in the institute to make a permanent flow into cancer research and so forth in MSK, in Weizmann. And so I think that this model could not be achieved otherwise. So the element of having the clinical doctors back into the very early research to have their own ideas, or I would say rephrasing our equation as basic research scientists into questions that are clinically relevant, which we could never do alone. And number 2, having a confluence of all the efforts into an objective that will have the best value for the money and also the highest speed.
So we are at the point of approval for prostate cancer. So that's happened very quickly. So we've run a large multi-center randomized clinical trial in Europe that proved that this therapy was effective at treating prostate cancer, and it's been approved by the EMA, and that therapy is now being approved or being evaluated for approval by the FDA. So we should know within this year about the approval status in the United States. Using funding from the Thompson Foundation, we also helped to run a phase 2 clinical trial at Memorial Sloan Kettering treating higher risk prostate cancer, which we just completed as of last week, actually. All 50 patients have been treated. Just to put things in perspective, the trial that was a multi-center trial run in Europe had 200 patients treated, and it took them 2 years to run that through about 30 different centers. At Memorial, we treated a 50-patient trial basically over about a 7 to 8 month period. So we were able to rapidly accrue patients to this study based on the strength of the therapy, which patients were certainly very interested in, but also the strength of the institution and the strength of the collaboration between Weizmann and Memorial. And when people knew about the science and they heard about it through our websites and through the Weizmann in terms of what this was all about, and realized how strong this was, this collaboration between the 2 institutions and what it could bring, patients came from all over. And I can't give you the results of the study. It's under lock and key until we have everything. But I would say that this therapy is incredibly promising, and I would be very shocked if by this time next year we're not talking about this as something as a standard of care, a normal therapy that we're giving to patients on a routine basis here in the United States. And I will tell you that now it's already starting up as a therapy in Europe. So, and all of that is possible. It would not have been possible without the help of the Thompson Foundation. And we tell them that all the time, and they tell us, that we're the crown jewel in their philanthropic program in terms of what they're doing. To start from what they fund and everything they do, and now to say they've developed a cancer cure for patients with prostate cancer is incredible. And where we are now with esophageal cancer, we're in the middle of that trial. I also cannot tell you the results of that study, although data was presented recently at a meeting showing that in these patients who have inoperable esophageal cancer. So to think about that for a minute in terms of what you're suffering from when you can't swallow, and someone tells you you can't have surgery, you can't have radiation, you're inoperable, you're intreatable. And then to suddenly treat somebody and find out that not only, this is the miraculous part of this that we don't talk about very much, but not only did this patient's cancer melt away in their esophagus, but shockingly, in a small number of patients, in a couple of patients, their metastatic disease also melted away. And that's part of the combination of this therapy with a new class of immune therapies, these immune point checkpoint blockade inhibitors that people talk about. You probably see advertisements for them, but when we combine these therapies together, we're seeing not only a local effect, but a systemic effect. That's, truly a miracle. And now we're looking at doing that in patients with bladder cancer and cancers that involve their kidneys, to allow these people to keep their organs functioning and allow us to treat not only the local extent of the disease, but metastatic disease as well. And so when you understand the true power of what this therapy can do, and know that the only reason why this is happening is because of the invention of this man, and all the other pieces that came together after that. And the weird part of that is, so I'll tell a little bit about, I won't take up too much time. I'm talking too much, I know I am. But the way he and I met was really interesting. I was at a meeting over in Europe, in Vienna. And I was giving a talk at the European Urologic Association meeting, and I'm down in the hotel, and in the gym, and I was on New York City time, so I'm down there, getting on the treadmill, and I'm on the treadmill, and there's this fellow next to me who's on the stepper machine. Elliptical. Yeah. He's very specific about the elliptical, and I can see he's struggling with this elliptical, and he's trying to figure out how it's working. And
oh no, I feel it.
Yeah. So I reached over and I just sort of said, I think you hit this button and you do this. And so he's like, okay, thank you. So then he starts doing his thing. And I'm sorry, Abby, do I have
No, it's okay. So it's a good story.
So he's going for a while, and then he's on his elliptical and I'm on and so he starts talking. And for those who know him, of course he's talking. So he's like, so what are you doing? And so I said, well, I'm here for a meeting. What do you do? And we go back and forth. And I said, I'm here for urology. Next thing I know, we're talking. And so then when we're done with our time, we sit down for a while. And I said, well, I'm working on this new type of therapy that we're working for prostate cancer. He said, really? I invented that therapy. And so I said, well, this is amazing. And I said, well, why are we doing this in prostate? All these other things we can do with it. And he said, yes. And we went back and forth. And it was that moment, where it just happened to be we were both in the same room at the same time. And that led to everything that's come after. And it's almost like this series of events that have occurred that have been tied together.
Serendipity.
Yes, serendipity, yeah. And it's like I said, it's like life's like a Plinko game. It just things happen.
Yeah, but it does need this kind of very rapid personal relationship, I must say, because there was an immediate chemistry. And it was clear to me. I think that trust and honesty and accountability are major parts of it. You make partnership with people that are accountable and that you can spend an hour. He hates it, but the bourbon, and you have your scotch. He's Scottish. But then there is the St. Andrews that both of us are with the scotch. So there is a beautiful friendship between the families. Jonathan was in Israel, at the Weizmann and in Tel Aviv, and the whole family. It's really to break so many barricades and things in order to know that you are together, that it is a togethership. And I think that this combination between the clinic and the basic research, the true combination, the respect, the patient to listen to each other, to rephrase the things, made it. That's.
I think at this point, I'd love to open it up to some of our guests here who might have a couple of questions for the doctors here. There.
Here you go.
Oh, we got a microphone for you there.
Thank you. Thank you, both of you, because you're able to affect a lot of people that we know that have these diseases. My understanding is that the treatment basically is for tumor-type cancers. So two questions. One, if that's the case, how does this particular treatment help with metastatic? That if the tumor has spread, how does that work? And can this also be used for pancreatic tumors?
Maybe you want to. Okay, so the idea is simple, but testing. It's now under real tested. It has been tested in animals successfully and is going now to the clinical trials, the next stage of our things. So here's the concept. The treatment comprises of sending a drug into the circulation, into the blood system. The drug stays in the circulation as a complex with serum albumin, which is the major protein in the blood, for a short time, relatively short time, about 60 minutes or so, and then it clears completely. Within that time, it's completely non-toxic in the dark. But as you shine light on it, it starts to make what we call radicals that are devastating and start a reaction that causes a collapse in a fairly selective way of the tumor, including all cells that comprise the tumor, cancer cells, immune cells, whatever. As these cells, and these radicals are very short-living, so the response is highly limited to the site of illumination, in particular to the tumor itself. There is a selectivity that was just shown by Jonathan in the clinical trials in the upper urinary tract. So once you break down these cells, it's like putting vaccination when you take bacteria and break them to pieces and let the immune system learn it. And the immune system learns the composition of these cells, the cancer cells, the other cells that comprise the tumor, whatever they are, immune cells and so forth. And then the immune system remember it and develop a response. That's what happened in the regular vaccination. So immune cells, toxic cells, start to look for other cells in the circulation, in the body, somewhere in the body that have the same stems as those that were just killed before, the same footprints. And these are other cancer cells that comprise the micrometastasis and later on the established metastasis. Now, what happened usually with tumors, with cancer, is that as the cancer evolve, it starts to enslave the immune system. What does it mean? The normal immune system, without any intervention, becomes all of a sudden pro-tumoric. Immune cells come into the tumor microenvironment, and somehow they talk with the cancer cells, and the cancer cells enslave them and teach them to become pro-tumoric. So we have to do 2 points. Number one, as I've said, while breaking these cells, we teach whatever has been left from the immune system, here is a danger signal. Go ahead and try to look for other cells like this. And at the same time, we add some materials that can be very cheap or more expensive, checkpoint inhibitors that you heard about, Keytruda and so forth, that will diminish this pro-tumoric effect. So this, by doing that, we move the scales into become anti-tumoric. So now we have an immune system that learn to recognize the cancer cells, and after breaking and destroying that local tumor that we could observe, in the prostate, in the lung, in the urinary tract, in the kidneys, in the breast, we now have an immune system that starts to surveil and look for other cancer cells to kill them. So this is how we go from the local therapy into the disseminated disease. Does it answer the question? Yes. Okay.
Another question. Gentlemen, thank you.
Question?
Richard.
In renal cancer, how do you illuminate without breaking the renal membrane?
So the question was, in kidney cancer, how do we illuminate without damaging the surrounding tissues or damaging the membranes inside? So kidney cancers come well, I would say cancers that involve the kidney come in sort of two different varieties. So there's one that is a tumor that involves the inner lining of the kidney, and then there's a tumor that involves the substance of the kidney. So when we think about kidney cancer, typically we're thinking about tumors that arise from the main substance or the the main organ of the kidney. So for kidney cancers, we can treat internally by giving the drug systemically, so it's an intravenous injection of the drug, and while the drug is circulating, we can place needles directly through the skin into the kidney to illuminate with light inside of the renal tumor, and that can destroy the tumor cells inside of the kidney that way. The other type of tumor that we treat is inside the renal pelvis, so one of the lining, or what I call the stomach of the kidney, where the urine collects. Those are the tumors that we're actually treating right now. And it's something that we call, it's called upper tract urothelial cancer. And that is a type of tumor that grows in that lining, in that stomach inside the kidney. So we do that by looking inside the kidney, just like you would do a colonoscopy, looking inside the colon. We can look up into the ureter and up into the lining of the kidney with a small scope we call a ureteroscope. And it's a very, very tiny scope, and through that scope we can pass a light catheter into the middle of the kidney. And so while we're staring at the tumor, you can actually see the cancer. It's kind of like growing like corals, like on a coral reef all around inside the lining of the kidney. And normally that patient would have their kidney removed. So the entire kidney would have to come out, the entire ureter would have to come out, and they would be down to one last kidney. But in these cases, what we do is we give the drug intravenously. We then illuminate inside the lining of the renal pelvis. And we can actually see the effects occurring inside the tumor. We can see some of the blood vessels start to diminish. But right away, nothing really changes. You know, you look at it when you're done, and you still see the tumor cells there, and you say, okay, things look pretty good. You leave, you come back a week later, and suddenly you see that all of the cancer that was there has just been completely obliterated. And the interesting part about this is, so we talked about prostate cancer treatment before. With prostate cancer, you can't see the tumor. So we're treating by putting the fibers into the prostate gland. And we put in several fibers inside the prostate gland, just like we talked about for the kidney before. We put them through the skin into the prostate. We give the drug, we turn the light on, we stop, we pull the fibers out, we're done. Here, a week later, I look into the kidney and the tumors are obliterated. I can see dead cancers sitting there. I can actually take out parts of the necrotic debris, which is there, the dead tumors. I can start to remove them. And this is where the science part comes in. This is where now I can take these tumors. Now, Avigdor has been treating tumors in mice over in the Weizmann Institute in Israel. And he's showing me all these great things he's doing in mice. And we're treating mice at our center, and we're sharing information back and forth. But now I have live tumor cells or dead tumor cells from a patient. And what we've been able to do is, in other models we've developed, we've taken tumors out of the kidney, we've pulled them directly out of patients, and we've put them into mice, and we've gotten them to grow. It's something called a PDX model, or patient-derived xenograft model. They're very expensive to run, but you can actually take patients' tumors and grow them right in a mouse and use that like we call it a fuzzy test tube. It's a little furry test tube with a tumor growing in it, and we can test drugs in that test tube, and we can test to see what happens. But we can use this therapy, 2CAT therapy, to show that we can kill cancer cells, human cancer cells that are growing in a mouse. And Avigdor's been doing that work with us, and he says, this is interesting, this is great, it looks wonderful. But now I can show him the actual tumor that we treated in an actual patient. And now we can start to look and we see, are the tumor cells dead? What have we done? What has been the mechanism of how this therapy has worked? Are the cells dying as we expect them to? Is the immune system being activated the way we expect it to? And so we can take that back to the lab. We can now learn so much from that now we can come back and say, you know what? We now have a way to maybe make this therapy better. We can combine it with this other therapy, or we can deliver the therapy in a new way and make it better. And that's what we call everyone talks about the bench to bedside approach, meaning we're taking it from the lab into the clinic. But it's the reverse effect that also is so important. It's the bench to bedside and bedside back to bench and then bench to bedside again. And this interaction is so incredibly important. And at Memorial, it's so important that we've actually built a huge building on 68th Street that is the Zuckerman building, that is this massive 20-floor building that has labs up and down the entire thing. But some of the expertise we have in that building is not the same expertise that they have at the Weizmann. And so being able to put our labs in contact with the Weizmann labs and my lab to work with his lab, and then Avidor to say, hey, we've got someone like Irit Sagi at our place who's doing this amazing work in tumor microenvironment studies. We should work with her. And then we get her to work with somebody at our center, and then next thing you know, we have this thing going back and forth. And suddenly you've got scientists working together in the same projects. Solving problems, which is what we're supposed to do. And it directly impacts patient care. But that's how we treat inside the renal pelvis, and that's how we kill those cancers, and that's how we save patients' kidneys.
So
I asked you the question, is 19 years ago, I had bilateral renal cell carcinoma.
Oh, you're kidding. Hope. So, but you still have a kidney, I hope, that's working.
3/4 of 1.
3/4 of 1. Today, we would be able to probably keep you with both.
So I just want to add 2 points here, which are just little one on what the wonderful thing that Jonathan, the first time that we really see the mechanism of action in working was 1 month after what Jonathan just described with this cancer in the upper urinary tract that completely vanished. And the urinary tract completely came to normal behavior. It looked like nothing happened in this area, no tumor, no stricture, normal urinary tract, it all works. This happened in most of the patients. Complete response. So this is one important thing, that we could never see the real theory work in action. The other thing referred to the question about the pancreas. Again, with the help of the Thompson Foundation, we established a huge program where this TUCA therapy is only one project within 3. Again, the unique about it is that the support of the Thompson Foundation actually enforced some scientists that would, before we just collaborate in conferences where everybody present his own achievement, like a bunch of men meeting in a hunting
And women.
Session, showing each of them show what he caught. But now they let everybody to learn from everybody, and we hope that with this we will be able to merge imaging and blood tests together so that when a patient will come at an earlier stage, the proper imaging technique will enable to find the tumor. There is the VTP, also TUCAD as a second project to treat the unresectable tumors. And then there is also part of it on the more advanced tumor. And we hope that we'll start to get clinical trials within a year and a half from now. We debate on the way to deliver the LiDAR. We try to find original lights. Again, Dr. Coleman is heavily involved there, as much as he does not want because he has such a huge burden, but his expertise is highly needed there, and there are some other agents. So it shows how, again, what Jonathan said, the knowledge of people that can do that and that, because at the Weizmann, we develop modalities that can be applied ad hoc in different ways of combinations to many different diseases. The bioinformatics, this TUCAD, MRI imaging that Lucio Friedman is developing, they can be applied to many different diseases. You have to define it. You have to congregate the people with the right clinician. And now you can find an original solution to unmet needs. And I think the whole idea here is actually going on unmet needs and finding solutions.
So there's a lot more to do. We're treating in 4 diseases. We've just started opening up a new avenue in pancreatic cancer. But there's a lot more, that just we're doing. It's a lot of heavy lifting in terms of trying to make all this work. At our place, and I think at the Weizmann, it's true too, that our goals are really to make sure that the patients come first and that the outcomes come first. And so, if someone comes to me and says, I have testicular cancer, I would not presume to treat that patient because it's not my specialty. I would refer them to somebody who does that. And the same is true. We're treating in pancreatic cancer at our place. We have leaders in pancreatic cancer at Memorial who are working in this process for pancreatic cancer. Leaders in esophageal cancer who are doing that. People have asked me, would I become involved? I say, well, it's not really my field. We need to find the best possible people to do this. And it's bringing those best possible people together that really makes all of this work. And But there is more. There's breast cancer that we want to work in. There are, we've talked about melanoma. We work with Jed Wolchuk in his lab, extensively to work we're doing work with him now in urothelial cancers to work on developing this technology for bladder cancer and upper tract cancer. We also want to work with him in melanoma. And the new thing, which is actually you'll hear for the first time, we haven't talked about it yet, lung cancer. So lung cancer, that's our next big target. And we are going to approach the foundation about lung cancer. But this all takes effort, work, and actually, I don't take any funding from this, so I don't take any salary from it at all. It's all, I provide my services for free. It's not because, I don't put time and effort into it. I put a huge amount of time and effort into it, but the bottom line is we want the money to go towards the research. And that's, unless the money's going there, then it doesn't do its job. And the unfortunate part is that if I get an NIH grant or a DoD grant, and I have them, I'm required to take money from that. I have to be salaried. They have to supplement my salary with that money, even though I would rather have it go to the research. It's required, that money has to pay for my time, and you can't just turn it around and give it back, to the institution. It has to go there. And with philanthropy, we're allowed to put the money directly to research and make sure that it works.
I think there's time for one more question over here.
A practical question. First of all, magnificent, absolutely magnificent, spectacular. We're honored to listen to this. Explain the process once this becomes the standard of care, the recognized standard of care. What's the process by which it rolls out into the world and becomes ubiquitous?
So that's where industry, comes in. So at Memorial, we've done a lot of work at developing things. I mean, there's no greater startup nation, obviously, than Israel. I mean, they've got more startups than anybody. There's a book about it, in fact, or several of them. So, but you need that industry partnership. So what we do is we do the beginning stuff. We develop the approaches. We develop the techniques and the procedures and how to do it. We develop the science behind it. Then we hand it off to an industry sponsor. So we work with Steba Biotech. They have it. They're going to commercialize it. They're going to run that whole side of that enterprise. It's up to them, to kind of push that part of things. Our job is then the next step, which is right now. In fact, we're working with them and saying, listen, you guys run the trials. You guys pay for the trials. You do all the stuff. We'll run them when we can do them at our place. But we want the tissue from those trials, the patients that are being treated. And patients are happy to say, look, you do what you want. You do a biopsy after the treatment. If you want to do a biopsy, you go right ahead if it helps further science. And that's what's beautiful about working with patients, that patients are so selfless. But we take that tissue and we learn from it, and then we make it into something better. And then we go back to the company and we say, look, we just figured out a way to make this better. And this is how you do it. I give it to Avigdor. Avigdor goes back to the Weizmann. He figures out how to make it better. He comes back to me. We share ideas back and forth. We talk with Cold Spring Harbor. We talk with the guys at Cornell. We have a collaborative team that works together. We make a better approach. We go back to the company and say, here, this is how it's better. And then they say, okay, we'll incorporate that now into what we do on the commercial side. But there's only so much, and we know what our roles are. That's the key part, is we do what we do best, and they take it from there.
So this is the actual final question here.
Yeah.
Yeah. When I heard you guys talk about a year ago, the two things that I found really interesting was the length of time of this procedure was relatively short, but also you had mentioned that there was someone who had gone through the procedure for prostate cancer, and literally the next day he was on a cross-country bicycle trip. The fact that you can have this procedure and life goes on as normal right after that, I thought was something that was really impactful.
So just to clarify, it wasn't the next day because you got to spend 24 hours out of sunlight, so you can't go out in the sun. But I will say, I actually had a guy recently who was treated, and he texted me 2 weeks after the procedure. He was helicopter skiing somewhere down in South America, and he showed me a picture of him getting out of the helicopter, about to go down, and he said, feeling great after my procedure from a couple of weeks ago. It took me a week to get to South America and to get up here, but I've been skiing all week and I feel wonderful. And he had this picture, and I thought, oh my God, he's out in this brilliant white sun with all this white stuff around him, something's going to happen. But in fact, I just saw him back last week for his 1 year, year and a half appointment. And I'll tell you, he's still cancer-free, and he's doing wonderfully, and he couldn't be happier. And so I wanted to know where he was going next. I said, where's the next, extreme adventure vacation he's going to be going on? But yeah, patients really can be back. I'm not going to say that these treatments don't have side effects. There are, when you put needles into someone's prostate gland, you're going to feel some side effects from it. But I mean, compared to what we see with other types of therapy, this is really, really miraculous.
Victor, Jonathan, we started off hearing Lester Crown talk about transformational philanthropy, and now we're talking about transformational medicine. So on behalf of all the guests tonight, the American Committee, thank you both so much for sharing your story.
Welcome.